So, imagine driving your car every single day for like 40 years.
Okay.
You rely on it to get to work, to get your family around safely. You turn the key, you step on the gas, and it just works.
Right.
But here is the catch. Absolutely nobody on the planet, not you, not the mechanics, not the engineers, actually knows how an internal combustion engine works.
Oh, wow.
Yeah, you just turn the key, watch the car move, and basically pray it keeps working.
I mean, it sounds completely absurd when you frame it around a car, but it's actually a brilliant comparison for the realities of early medicine.
Because if you swap out that car for the most famous antibiotic in human history, penicillin, that is exactly what happened.
Yeah, it really is.
They mass-produced it, they gave it to millions of soldiers and patients and saved, countless lives during and after World War II.
Unbelievable numbers.
But the crazy thing is, nobody on Earth actually knew how penicillin worked at a molecular level for nearly 4 decades.
4 decades of flying totally blind.
Exactly. And that blind spot, that mystery of how this microscopic war is actually fought inside your body, is exactly what we are focusing on for today's deep dive.
It's a huge topic.
It is. We have a fascinating stack of microbiology research papers and medical texts right here in front of us, and they are all centered on one incredibly vital concept: antimicrobial mechanisms of action.
Which is, truly the ultimate look behind the curtain. Because when you break a bone, the X-ray shows a jagged white line.
Right.
It's binary, it's visible, and it's somewhat comforting. But when you swallow an antibiotic, you are initiating this highly complex, completely invisible biochemical assault.
And our mission today is to look beyond that literal pill bottle. We want you to understand exactly what happens at a microscopic scale when that drug hits your bloodstream.
Step by step.
Yes, we are going to break down the 5 specific ways modern medicine can stop a microbe in its tracks. We'll explore why these so-called magic bullets sometimes completely fail, and ultimately, why understanding this molecular arms race is crucial for your own health.
Because it affects all of us.
It really does. Okay, let's unpack this, starting with what a mechanism of action actually is. Because in the medical world, it's a very specific, fiercely guarded term.
Yes, it is. It is not just about what illness a drug treats. That's its spectrum.
Right.
And it's not about how your body absorbs or digests the pill, which is pharmacokinetics. The mechanism of action is exclusively about the physical, biochemical reality.
Like, what is the drug physically doing to the bacterial cell?
Exactly. It is the molecular how, what target does it bind to, and what biological cascade does that trigger.
Right.
But before we can dive into the specific targets of today's drugs, we have to establish the philosophical foundation that makes any of this possible.
The golden rule of pharmacology: hitting the bug without hitting the patient.
You got it. The scientific term for that is selective toxicity.
Selective toxicity.
And this concept was championed at the turn of the 20th century by a German physician-scientist named Paul Ehrlich. He actually coined the term chemotherapy.
Oh, I didn't know that.
Yeah, and he had this beautiful, almost poetic phrase for what he was looking for. He called it Zauberkugel, or the magic bullet.
It sounds like something pulled straight from a fantasy novel.
It really does.
But his logic was ruthlessly pragmatic. I mean, he theorized that if you could find a physical structure or a chemical process that exists in the invading pathogen but is completely absent, or at least very different, in the human host, you could exploit it.
Exactly. You could poison the invader without harming the patient.
And Ehrlich actually put his money where his mouth was, right?
He did. He methodically tested hundreds of arsenic-based compounds against the spiral-shaped bacteria that causes syphilis.
Wow, hundreds.
Yeah. And finally, in 1910, his team discovered compound 606, which was later marketed as Salvarsan. It was toxic to the bacteria, but relatively tolerable for the patient.
Setting the template for literally every drug discovery program since.
Absolutely.
And the history here is just full of these incredible high-stakes moments. You have Gerhard Domack in Germany in 1932. He's desperately testing this red industrial dye called Pronticel.
Such a wild story.
It is. He actually ended up using it to cure his own daughter, who was dying from a severe streptococcal infection after pricking herself with a sewing needle.
I mean, the desperation of that moment.
Truly. And it turned out Pronticil was what we now call a prodrug. The body breaks the dye down into an active compound called sulfanilamide, which became the world's very first anti-metabolite antibiotic.
A completely harrowing story with a miraculous outcome. And of course, that brings us to the most famous happy accident in scientific history.
Alexander Fleming.
Right. September 1928. He returns from vacation to St. Mary's Hospital in London, examines a contaminated petri dish of Staphylococcus, and notices this clear ring where no bacteria can grow near a mysterious patch of Penicillium mold.
But the part that often gets glossed over is that for over a decade, Fleming's mold juice was just a laboratory curiosity.
It was notoriously unstable.
Yeah, it wasn't until Howard Florey and Ernst Chain at Oxford finally managed to purify and mass produce it that it changed the world. Which brings us back to our car analogy. Because they are pumping this miracle drug into millions of people, and it works, but they are flying blind.
Totally blind.
It wasn't until 1965 that researchers Tipper and Strominger finally mapped the actual biochemical reality.
They finally cracked the code.
Yeah.
Tipper and Strominger discovered that penicillin's molecular structure, specifically this square-shaped core called a beta-lactam ring, was essentially a chemical disguise.
A disguise.
Yeah, it perfectly mimics a specific molecule found only in bacteria, a structural building block called the D-alanine terminus.
Wow.
And by mimicking this block, penicillin violently jams the machinery the bacteria uses to build its protective outer shell. If we connect this to the bigger picture, that 1965 revelation is the ultimate vindication of Ehrlich's magic bullet.
Because it targets something we don't even have.
Exactly. Penicillin works entirely because human cells do not use D-alanine. We don't have the machinery it jams. Every single antimicrobial mechanism we're about to discuss relies on this exact principle of selective toxicity.
We are always hunting for a biological target that the bacteria absolutely need to survive, but that we just don't have.
Precisely.
That perfectly sets up our battlefield. If we think of a bacterium as like a microscopic medieval castle we're trying to siege, the sources break our attack strategies down into 5 classic targets.
Let's do it.
Let's start with the outer defenses. Target number 1, cell wall synthesis.
So human cells are basically squishy balloons. We just have a flexible lipid cell membrane.
Right.
But bacteria are dealing with immense internal fluid pressure. To keep from popping, they encase themselves in a rigid chain-link mesh called peptidoglycan.
It is essentially their suit of armor.
Exactly. And drugs like penicillin or other beta-lactams like cephalosporins go after the specific bacterial enzymes that cross-link and stitch that armor together.
Those enzymes are actually called penicillin-binding proteins, right?
Right. And when the drug permanently binds to them, the armor can't be stitched together, and the sheer osmotic pressure inside the bacteria causes it to quite literally burst open.
Just a catastrophic structural failure. Now, there are other ways to attack that same wall, and I really want to bring in an analogy here because the difference between how penicillin works and how another heavy hitter drug, vancomycin, works is just fascinating.
It's a great comparison.
So let's imagine building that peptidoglycan wall is like stapling thick stacks of paper together.
Okay.
The bacterial enzyme is the stapler. Penicillin works by flying in and physically breaking the stapler mechanism itself.
Highly effective. The enzyme is neutralized.
But vancomycin, which is a massive molecule called a glycopeptide, operates one step earlier. Instead of breaking the stapler, vancomycin flies in and slaps a thick, impenetrable piece of tape over the exact spot on the paper where the staple is supposed to go.
In chemistry, this is called steric hindrance. It physically crowds the space.
Right. Vancomycin binds directly to that D-alanine building block we mentioned earlier, capping it off so the stapler can never even reach it.
It's the exact same goal of stopping the wall, but through an entirely different molecular mechanism.
That is just so cool.
It is a phenomenal way to visualize it. And because human cells have 0 peptidoglycan, both the broken stapler and the taped paper methods are beautifully selectively toxic to the bacteria.
Which naturally leads us to the second barrier. What happens if we bypass the rigid wall and go after the squishy moat underneath it?
Target number 2, cell membrane disruptors.
Right. But to understand this one, we have to quickly clarify a major division in the bacterial world. Gram-positive versus gram-negative bacteria.
A very important distinction.
Gram-positive bacteria have a super thick peptidoglycan wall on the outside, but gram-negative bacteria have a much thinner wall sandwiched between 2 separate lipid membranes.
They have that extra outer shield.
Yeah, and that outer shield is incredibly tough to penetrate.
This is where drugs like the polymyxins, specifically colistin, come into play. Colistin acts almost like a molecular detergent.
A detergent, wow.
Yeah, it binds to the outer membrane of gram-negative bacteria and forcibly kicks out the calcium and magnesium ions that normally stabilize the structure. The membrane loses its integrity, becomes porous, and all the vital cellular contents just leak out.
But reading the source material, there is a massive red flag with colistin. It flirts dangerously closely with the boundaries of selective toxicity.
It really does.
Because our human cells are also bounded by lipid membranes, colistin can sometimes be indiscriminate, leading to severe toxicity in human kidney cells. I read that doctors actually shelved it back in the 1970s because it was deemed just too dangerous.
They did. But the tragic irony is that in the 2010s, hospitals were forced to pull it out of retirement.
Because of resistance.
Exactly. The rise of multi-drug resistant superbugs meant that for some severe gram-negative infections, colistin became our absolute last resort. You have to risk the kidney damage because the alternative is certain death from the infection.
That is a terrifying trade-off.
It is. Now, on the flip side, for gram-positive bacteria, we have drugs like daptomycin, which inserts itself into their single membrane and causes a massive, rapid loss of potassium ions, essentially short-circuiting the cell.
So we've breached the outer wall, we've poisoned the moat. What happens when we get inside the cell?
We attack the internal machinery.
Exactly. That brings us to target number 3, protein synthesis, the ribosomes.
Right, so every living thing needs to manufacture proteins to survive. And ribosomes are the microscopic factories that read genetic instructions and assemble those proteins.
We have them, and bacteria have them.
But again, Ehrlich's magic billet saves us. Bacterial factories are structurally distinct. They are classified as 70S ribosomes, made of a 30S and a 50S subunit.
Okay.
Human cytoplasmic ribosomes are slightly larger and denser, classified as 80S. And that tiny structural gap is all the space a clever drug needs.
And the ways these drugs jam the factories are incredibly specific. Let's take aminoglycosides like streptomycin. These drugs bind to the smaller 30S subunit and essentially warp its shape. The factory doesn't stop, but it starts misreading the genetic blueprints.
Oh, that's clever.
Right. It starts churning out faulty garbage proteins that build up and become toxic, eventually killing the bacteria.
And then you have the tetracyclines, which also hit the 30S subunit, but they simply block the delivery trucks, the transfer RNAs, from bringing new amino acids to the factory floor.
The assembly line just halts.
Exactly. But the one that really stood out to me was the macrolides. If you've ever taken a Z-Pak or a zithromycin for a nasty sinus infection, this is what you were doing.
Oh, I've definitely taken a Z-Pak.
Most people have. Macrolides target the larger 50S subunit. If you picture the ribosome as a 3D printer churning out a long plastic chain, the macrolide acts like a giant cork jammed right into the exit nozzle.
Wow.
The printer reads the instructions, links a few amino acids together, but the chain has nowhere to go. The entire factory jams up and shuts down.
It is a stunning level of molecular interference. Moving even deeper into the cell, past the factories, we reach the command center. Target number 4, nucleic acid synthesis.
Hitting their DNA and RNA directly.
Yes. This is where drugs like the fluoroquinolones come in, like ciprofloxacin or Cipro, which a lot of people take for severe UTIs.
Very common.
The mechanism here absolutely blew my mind. When a bacteria wants to replicate, it has to unzip and copy its massive circular strand of DNA.
Right.
But as it pulls the strands apart, the DNA ahead of the zipper gets tangled and tightly wound up like when you pull apart a twisted rubber band.
The scientific term for that is supercoiling. And if the tension isn't relieved, the replication machinery stalls and the DNA can actually fracture.
So to survive this, bacteria use specialized enzymes, DNA gyrase and topoisomerase DV, to temporarily cut the DNA, let it spin to release the tension, and then seamlessly glue it back together.
Which is an incredible process on its own.
It really is. And Cipro works by sneaking in and jamming the gluing mechanism. Yeah, the bacteria cuts its own DNA to relieve the tension, but Cipro prevents it from repairing the cut. The bacteria essentially shreds its own instruction manual.
A completely catastrophic event for the microbe. And again, human cells have topoisomerase enzymes too, but their physical shape is different enough that Cipro just ignores them.
Magic bullet again.
Exactly. You also have drugs like rifamycin, which block bacterial RNA polymerase from transcribing DNA into messages, which is vital for treating tuberculosis because rifamycin can actually penetrate the waxy outer shell of mycobacteria.
Okay, so we have the wall, the membrane, the protein factories, and the DNA command center. That brings us to the 5th and final classical target. Metabolic pathway inhibitors, the antimetabolites.
A very elegant strategy.
It might be my favorite because it's so strategically elegant. We are essentially starving their supply lines.
Specifically, their supply of folate or vitamin B9.
Right.
Both humans and bacteria desperately need folate to synthesize the building blocks of DNA and RNA. Without it, replication is simply impossible.
But here's the vulnerability. Humans get our folate by eating it. We absorb it from leafy greens and our diet.
Right.
Bacteria cannot absorb it from their environment. They are biologically forced to manufacture their own folate from scratch, using a specific assembly line of enzymes.
Which leaves them incredibly exposed. If we block that assembly line, they starve, while our human cells just keep happily absorbing folate from our lunch.
We do this using a two-pronged attack. We use a class of drugs called sulfonamides to block an early step in their folate manufacturing line.
Okay.
Then we use a drug called trimethoprim to block a later step in that exact same line. When you put them together in a pill most people know as Bactrim, you hit the bacteria with a sequential double blockade.
It's a textbook example of pharmacological synergy.
Yeah.
By hitting two steps in the same vital pathway, the combination of the two drugs is exponentially more effective than either drug acting alone.
So we have our 5 highly specific targets. Now that we understand exactly what these drugs are looking for, it makes it incredibly obvious why antibiotics are totally useless against viruses.
Oh, the classic misconception.
Right. I have a terrible viral cold. I demand a Z-Pack. Mechanistically, that is just doomed to fail.
Just run down the checklist we just built. Viruses do not have a peptidoglycan cell wall for penicillin to break.
Yep.
They don't have their own ribosomes for a Z-Pack to jam. They invade your cells and hijack your human ribosomes.
Right.
And they lack independent metabolic pathways to starve. Prescribing a bacterial cell wall inhibitor for a virus is like bringing an anti-tank missile to a submarine fight. The targets simply do not exist.
And worse, taking that drug just puts evolutionary pressure on the good bacteria naturally living in your gut, breeding resistance without doing a single thing for your viral throat infection.
Exactly. It's all risk and zero reward.
So, what does this all mean for how we actually measure the success of these drugs? Our sources spend a lot of time drawing a hard distinction between drugs that are bactericidal, meaning they cause the bacteria to burst or shred its DNA and die outright, and bacteriostatic, meaning they just jam the factories and stall the bacteria's growth.
A very important clinical distinction.
But my immediate instinct is, isn't a dead bug always better than a stunned bug? Why would a doctor ever prescribe a drug that just stalls the infection?
It's a completely natural assumption. But the kill versus stun framing doesn't dictate clinical reality.
Really?
Yeah. If you are a relatively healthy person with a functioning immune system, a bacteriostatic drug that merely halts the bacteria's replication is often exactly what you need.
Because it gives your body time to catch up.
Exactly. Once the bacterial population efficiently clear the infection.
So we're basically just holding the enemy down while the immune system delivers the knockout punch.
Beautifully stated. Bactericidal drugs, the outright killers like Cipro or penicillin, are strictly necessary when that immune punch isn't guaranteed.
Ah, okay.
Like if a patient is immunocompromised, or if the infection is hiding in a protected site that white blood cells can't easily patrol, like an infection deep inside a heart valve, then you need a drug that finishes the job entirely on its own.
That makes perfect sense. Another huge misconception the texts point out is the concept of antibiotic resistance itself.
People get this one wrong a lot.
They really do. People hear the drug stopped working, and they picture a bottle of pills sitting in the cabinet losing its potency, like the drug itself is chemically degrading.
That is a very dangerous misunderstanding. A vial of penicillin from 1950 is chemically identical to a vial today.
Right?
Resistance is an evolutionary property of the bacterial population, not a flaw in the drug. When subjected to the pressure of an antibiotic, the surviving bacteria actively mutate.
It's survival of the fittest.
Exactly. They physically alter their internal targets, or they build microscopic pump systems to spit the drug back out, or they synthesize brand new enzymes to destroy the antibiotic on contact.
Which transitions us to the modern crisis, the evolutionary arms race. And the wild part is, none of this was a surprise. Alexander Fleming literally called it from the podium when he accepted his Nobel Prize.
He did. In his 1945 Nobel lecture, he issued a completely chilling warning.
Yeah.
He described a hypothetical patient he called Mr. X. Mr. X buys penicillin, but he doesn't take the full course. He underdoses himself.
A huge mistake.
Massive. Because the concentration is too low to wipe out the infection, the most robust bacteria survive the exposure. Mr. X unknowingly breeds a hardened, resistant strain in his own body, infects his wife with it, and she dies because the penicillin can no longer save her.
A completely prophetic warning. I mean, within a decade of that speech, hospitals were overrun with strains of Staphylococcus that had evolved to produce penicillins.
Right.
It's an enzyme that acts like molecular scissors, literally cutting open the beta-lactam ring of the penicillin before it can even reach the cell wall.
And that arms race has only accelerated into highly sophisticated countermeasures. Look at vancomycin, the heavy hitter we discussed that puts chemical tape over the dealanine target.
Right, the tape on the paper.
Yeah, it was introduced in 1958 and served as our reliable, unbreakable last line of defense for decades.
Here's where it gets really interesting. In 1986, researchers discovered the first vancomycin-resistant enterococci, or VRE.
A huge blow to modern medicine.
Remember how vancomycin specifically binds to the shape of that dealanine handle?
Yes.
Well, these resistant bacteria evolved complex genetic machinery that allowed them to swap that handle out for a slightly different molecule, D-alanyl D-lactate.
It is a microscopic evolutionary heist.
It really is.
By changing one single atom at the end of that chain, swapping a nitrogen for an oxygen, the bacteria altered the physical shape of their cell wall just enough that the massive vancomycin molecule simply slips right off.
Wow.
The drug's entire mechanism depends on a perfect steric fit, and the bacteria just change the locks.
And the stakes are only getting higher. The source material highlights a terrifying turning point from 2015 in China.
The discovery of the MCR1 gene.
Yes.
This is arguably one of the most alarming public health moments of the modern era. Researchers found a gene that confers absolute resistance to colistin.
And remember, colistin is the membrane-destroying drug that is so toxic to human kidneys, we only use it as an absolute last resort for gram-negative superbugs.
Right, so losing it is devastating.
But the really chilling part wasn't just that the resistance existed, it was how the gene was packaged.
Precisely. The MCR1 gene was found riding on a plasmid.
Okay, explain that for us.
So normally, bacteria pass down genetic traits vertically, from parent to offspring, when a single cell divides. But a plasmid is a small mobile loop of extra DNA that bacteria can trade horizontally with their living neighbors.
Wait, horizontally?
Yes. They can literally hand the instruction manual for colistin resistance to a completely different species of bacteria floating right next to them.
It's like sharing an invincible cheat code over Bluetooth.
That is exactly what it is.
Yeah.
And because it is so easily transferable across species, this exact gene was detected on multiple continents within a matter of months.
Oh my gosh.
Yeah. It raised the very real specter of truly untreatable pan-resistant superbug infections spreading globally.
It's terrifying, but it also highlights how incredibly dynamic and unresolved this field of study really is. Even the rigid textbook rules we laid out earlier aren't totally set in stone.
What's fascinating here is that even the neat 5-category model of targets is actively debated at the frontiers of science.
Really?
Oh, yeah. In 2007, James Collins' lab at Boston University published a highly provocative paper. They looked at 3 totally chemically unrelated classes of bactericidal drugs: beta-lactams targeting the wall, aminoglycosides targeting the ribosomes, and fluoroquinolones targeting the DNA.
Okay.
And they proposed that they all actually share a common downstream killing pathway.
Like a common mechanism of death.
Yes. They argued that regardless of their primary target, all of these lethal drugs eventually trigger a metabolic crisis that floods the inside of the bacterial cell with toxic hydroxyl radicals.
So extreme oxidative damage.
Exactly. It's oxidative damage that ultimately scrambles the inside of the cell and delivers the fatal blow.
Meaning that focusing solely on the 5 neat little categories might severely undersell how complex the death of a bacteria actually is.
It was a massive paradigm shift. Now, to be fair, this hypothesis received substantial pushback.
Naturally.
Other researchers argued the oxidative stress was a minor side effect,
Oh.
or heavily dependent on how the experiments were run in the lab. The debate rages on today.
But it is a perfect reminder that mechanism of action isn't just settled, dusty history to be memorized. It is an active, bleeding-edge frontier of science.
Absolutely.
And that brings us full circle to why any of this matters to you, listening right now. Understanding antimicrobial mechanisms isn't just trivia for medical board exams. It is the direct basis of how doctors choose empiric therapy.
Guessing the right drug before the lab results even come back.
Right. It is how the World Health Organization prioritizes the greatest global biological threats. And it dictates how researchers desperately try to design the next generation of drugs.
Because bacteria evolve resistance faster than we can bring new molecules to market.
Yeah, nearly every new antibiotic class has to find an entirely novel molecular vulnerability to exploit.
It is an endless race.
Okay.
But understanding the mechanisms gives us a map of the battlefield.
It really does.
Speaking of which, thinking about how these bacteria operate leaves me with a somewhat provocative thought to end on.
Oh, let's hear it.
We just talked about how terrifying it is that bacteria share resistance genes like that MCR1 plasmid horizontally. They casually trade these genetic cheat codes to turn harmless neighboring bacteria into invincible superbugs.
Right.
But if bacterial populations are so inherently eager to trade genetic code with each other, could scientists theoretically weaponize that exact system?
Wait, what do you mean?
Could we engineer harmless synthetic bacteria designed to horizontally transfer genetic vulnerabilities or even dormant self-destruct mechanisms back into the superbug populations?
Oh, wow.
We could essentially use their own communication network, their own genetic Bluetooth, to disarm them from the inside out, turning their greatest evolutionary advantage into a Trojan horse.
That is a wildly compelling concept, and definitely something to ponder next time you look at a simple plastic pill bottle.
Yeah.
We started today talking about how much we love the certainty of a broken bone on an X-ray, the comfort of things we can easily see. But as we've explored, the real fight for our survival isn't visible at all.
Not even close.
It is a microscopic, shifting chess match of enzymes, proteins, and evolutionary heists happening inside you every single day. Thank you for joining us on this deep dive into the unseen, and keep asking questions.